Welcome to the Defeating Epilepsy Foundation YouTube channel. In this presentation, we will discuss what SPTAN1 is and its functions, the etiology of epilepsy in SPTAN1, symptoms, diagnosis, and treatment options. Make sure to like our videos and subscribe to our YouTube channel. Click on the bell to be notified of future videos. Please click on our donate link in the description section below and donate today. Your donation helps us to make a difference for those battling epilepsy. We appreciate your support. SPTAN1 is a crucial gene that provides instructions for making alpha-2 spectrin, a cytoskeletal protein abundant in the brain. This presentation will explore the role of SPTAN1 in epilepsy, its symptoms, diagnosis methods, and potential treatments. We'll delve into the complex world of genetic mutations and their impact on neurological function. SPTAN1 is a spectrin, a family of proteins playing a crucial part in the cell membrane that provides instructions for making a cytoskeletal protein called alpha-2 spectrin, which surfaces on the plasma membrane of eukaryotic cells. It is abundant in the brain and plays a crucial role in the formation and maintenance of cytoskeletons, network of protein filaments, thread-like structures, and tubules, tube-like structures, that extend throughout the cytoplasm of eukaryotic cells, in neurons. Mutations in the cytoskeletal protein, the SPTAN1 gene, have been viewed in starting epileptic encephalopathy and structural abnormalities contributing to the onset of epilepsy. For epileptic encephalopathy to occur among infants and younger children, a heterozygous mutation occurs in the SPTAN1 gene. There have been many cases leading to the identification of a non-heritable heterozygous mutation in the SPTAN1 gene. Studying the expression of the non-heritable heterozygous mutation, harmful gene products interfere with the parts, spectrins, causing normal structural stability in non-covalent interactions of the cell membranes, particularly in red blood cells. This leads to decreased stability in the cell membrane, making it lose shape, integrity, and causing it to be susceptible to stress. The non-heritable heterozygous mutation in the SPTAN1 gene also can disrupt the spaces between segments in the axon, causing functional changes to the neuronal charge in the brain. Along with this disruption, it may also be followed by only one copy of the SPTAN1 gene, instead of two copies, and accumulations of spectrins. This causes epileptic encephalopathy through hyperexcitability of the neuronal charge and other effects. Symptoms of SPTAN1-related epilepsy are hypsarrhythmia, lack of visual attention, poor head control, feeding difficulties, microcephaly, spastic quadriplegia, cerebral atrophy, and hypomyelination. It is most often difficult to diagnose mutation in the SPTAN1 gene. There are some ways, however, that have helped identify mutation in the SPTAN1 gene. MRIs have been effective in finding a mutation in the SPTAN1 gene through the identification of brain atrophy, detailed to be a symptom of the mutation. There is a reduced volume of white matter, atrophy/hypoplasia of the brainstem and cerebellum, and among other abnormalities. Protein contact maps have also been effective in finding mutations in the SPTAN1 gene through amino acid residues. Double immunostaining found that certain protein combinations, specifically alpha-2 and beta-2 and alpha-2 and beta-3 spectrins, were involved in clumping together, aggregation. A mutation called P.Q2202del didn't change the pattern in N2A cells, but in brain cells, it still caused aggregation, though in fewer cells. The findings suggest that the severity of symptoms might be linked to how much these proteins clump together. Interestingly, two patients with a certain gene duplication didn't live long, while another patient with a different mutation survived longer despite severe movement problems. Although there are little to no studies looking at therapy specifically for SPTAN1 gene, there have been recent developments in targeting pathogenic variants in spectrin including the SPTAN1 gene, potentially leading to therapeutic intervention. Spatiotemporal mapping of spectrins has been helpful in evaluating the progression of spectrin, therefore providing when to treat for pathogenic variants of spectrin such as SPTAN1 gene. In addition, the reintroduction of spectrin through animal models has allowed researchers to evaluate the pathological mechanisms involved in pathogenic variants, allowing researchers to know where and the cause of the pathogenic variants. When coupled with genomics and other impartial high-throughput molecular approaches, this comprehensive toolkit will pave the way for pinpointing targets through conventional therapeutic exploration, an area currently lacking in the study of spectrinopathies. Consequently, these tools will facilitate the adoption of promising technologies that could provide alternative, viable routes for treatment, including gene replacement and editing, as well as strategies based on antisense oligonucleotides 182, short sequences of nucleotides designed to specifically bind to complementary RNA sequences. Challenges in treating SPTAN1 gene mutation is due to limited studies, complex mechanisms, personalized treatment for patients, and ethical considerations. As our understanding of SPTAN1 and its role in epilepsy grows, we move closer to developing targeted therapies and improving outcomes for affected individuals. Continued research and collaboration will be key to unlocking new possibilities in treatment and management of this complex condition. To learn about SPTAN1 gene mutation, please check out the resources used in the presentation today. Please like and subscribe to our YouTube channel and follow us on our social media pages. We would love to hear from you. Leave a comment below or email us at info@defeatingepilepsy.org. Thank you for your support, and together we will defeat epilepsy. Check out our other great videos and subscribe today. You have the power to defeat epilepsy.